Now the question arises, how can you see the red blood cells under a microscope? Well, for that we need to make a very thin film that we call as a smear, right? So what do we need to make? We need to make a very thin film on a glass slide that we call as blood smear. So let's see what do we need to do for making a blood smear. Just look in the image. First of all, you need to have a drop of blood, and for that just take a sterilized needle, prick it on your finger. Please just wipe off the first blood that comes out. The blood that comes next, put that drop of blood on the right hand side of the glass slide. Just look in this image, and in just this way you have to do it. So place a small drop of whole blood on a very clean slide and hold a second slide at this angle, and this angle is approximately 60 degrees. So hold this second slide at an angle of 60 degrees. So while maintaining contact with the bottom side, pull the top slide back to contact the top, which will spread by capillary action. You can see that the blood is going to spread by a capillary action. Now after this, maintain firm contact with the bottom slide and push the top slide in one motion to produce the smear, right? After this, just push the slide in one motion. You don't have to stop in between, just push it in one motion to produce the smear. Now let the smear dry for some time. After the smear dries, just put a stain that we call as Leishman stain. Put Leishman stain on it, and after some time put distilled water and then let the slide again dry. After this, you have a slide which has this blood smear which is going to show you the red blood corpuscles, and this is also going to show you this under high magnification, right? So just remember how you're going to make a blood smear and how this actually is going to happen. So you need what? You need two clean glass slides. You need a sterilized needle so that you can prick your finger and you can have the drop of blood. Just remember that put it on the right side and take another slide, just that it comes in contact with the blood drop. When it comes in contact with the blood drop, what is going to happen? The blood drop is going to spread itself by capillary action, and you just have to maintain that firm contact. You don't have to press it hard, just maintain a firm contact with the bottom slide and push the top slide in one motion. And after this, you will have a thin film of blood which we call as blood smear. You can see this slide under microscope, and this is how you can have a blood smear. Just stain it with Leishman stain so that you have more clear visibility of the red blood cells, and you can see it under high magnification.